Disclaimer: as a result of continuous updates to the bioinformatics web pages, you may find that the web pages and the video are not exactly the same as what you see on your own screen. Please refer to your exercise sheet or PowerPoint slides for most up-to-date information. In this video, we will learn how to translate the nucleotide sequence, compare the nucleotide sequence query against bioinformatic databases to find the gene of origin or genes with similar sequences, and learn how sequence data is represented in FASTA format. First, I will demonstrate how to retrieve nucleotide sequence. Go to bioinformatics.org to reverse transcribe your amino acid sequence. Paste your sequence and hit Submit. This window will pop up. Choose only one, usually the first reverse translated sequence out of the many. So this is the first one for me. I will copy that. Now go to BLAST. BLAST is a collection of programs that compares a sequence of interest against a database of sequences. It checks to see which sequence are most similar. We will be running the nucleotide BLAST program, which compares a nucleotide sequence against nucleotide sequences in a database. Click on Nucleotide BLAST. Paste the nucleotide sequence that you just copied, leave all the parameters as it is, and then hit BLAST. Note, if you do not get the results, then in the selection section right here, you can select a different option. Hit BLAST. Note, this may take a while. On top left of the page, under the nucleotide sequence, you will find information about the query, including RID, program, molecule type, query length, etc. RID is the job identifier used to return to the search query. Note, this ID would be different after each run. Below, a list of match sequences producing significant alignments sorted by eValue, person identity, accession, etc. will be listed. The top pick is the gene with very high degree of certainty. Now, if we go back up and click on the citations, we will be able to see the reference here. Now, click on the first entry. Then click on the GenBank. This will bring you to the GenBank page. It will provide you the information about the base pairs, organism, etc. You can also find the PubMed ID in here. Click on the FASTA link. Here, you can copy the first two lines of the sequence. From the previous page, go back to descriptions and click on the accession for the first entry. Accession is the unique identifier for a particular sequence entry. It will take you to the NCBI GenBank page. GenBank being the database of the gene, we can find more information about the gene. Locus, right here. Base pairs, source, etc. Other details can also be found on this page. You can either scroll down or use Ctrl F feature. Depending on the type of sequence, you may find a note description of the gene and links to the other databases, as well as amino acid translation of the given DNA sequence. Note, unlike human protein, Helicobacter pylori contains the whole genome. Thus, you may or may not find the information about the locus or chromosome if your protein is from a species other than humans. On the NCBI entry page, click on FASTA link. FASTA stands for Fast All or FASTA. It represents nucleotide or amino acid sequences. It contains information regarding the sequence along with containing the sequence itself. Here you will find the descriptor of the sequence in the first line, followed by the sequence itself. Now we will try to look for open reading frames. It is a continuous stretch of codons that is actually translated. It begins with a start codon and ends with stop codon. To find open reading frame for your sequence, go to NCBI's ORF Finder. In the search box, you can enter the GenBank accession number. I have already pasted it here, but you can also find it if you go back to the NCBI page. And here is the GenBank accession that you can copy and paste. Leave all the parameters as it is, and then hit Submit. You will find many possible open reading frames, but generally the longest one is the coding sequence. For us, we can see the longest one is ORF 76. If you scroll down, you will see a table right here, categorized as label, frame, link, etc. You will be able to find both nucleotide and amino acid length for the ORFs using this table. Now, to know about the interactions the gene is involved in, the pathways or the functions, we can use pathway commons and gene card. First, we are going to use pathway commons to find the interactions our gene is involved in. Pathway Commons is a database that collects data from the partner databases. It includes biological pathway interactions. Search for your protein, urea, and then hit search. The total number of pathways will be shown in parentheses. In our case, we have a total of 85 pathways listed down here. Now, go to genecard.org. Type your gene ID or gene name in the search bar and hit search. For demonstration purposes, I will be using BRCA1 gene. On this page, you will find the information about the genes with which your gene interacts. Click on the hit, which has the maximum score. So if you look at the score, the first hit has the maximum score. On this page, you will find the gene card symbol, name, NCBI gene link or the external link, HGNC link, function, etc. Now we will go to Signor. There are two ways. You can either scroll down on the gene card page until it shows the Signor section, or you can go to the Signor website itself. Signor is a signaling network database that shows the interactions between biological entities as either upregulating or downregulating. Search your gene through Signor. For demonstration purposes, I will be using BCHE. Hit search. When you hover your mouse over Get Relations, you will be able to see the preview details. Now click on Get Relations. On this page, you will have Summary, Viewer, and the Relations table. Now click on Inhibitor or Regulator or Ligand, whichever one the summary section shows. So for me, it is Inhibitor. So I'm going to click on that. And it will bring you to the PubChem. Here, the structure is shown. That concludes this video. Hopefully, now you will be able to translate the nucleotide sequence, compare the nucleotide sequence query against bioinformatic databases, to find the gene of origin or genes with similar sequences and describe how sequence data is represented in FASTA format. The project is made possible with funding by the Government of Ontario and through eCampus Ontario's support of virtual learning strategy. This resource was created at University of Windsor under the supervision of Dr. Zareen Amtel.